Bacteria. They are on top of us, they are inside of us. Human and animal bodies host more than 100 trillion bacterial cells. There are more of them than there are cells of us. Bacteria coexist in vast microbial ecosystems. We refer to this as the microbiome. The microbiome offers many important functions to hosts, affecting brain health and mood, metabolism, digestion, and immunity against harmful organisms, and so much more. A key to a beneficial microbiome is a microbiome diverse in the types of different bacteria that reside. Microbiomes consisting of genetically different bacteria are more resilient to drastic or harmful changes. There is, however, a cost to this diversity. Diversity is maintained by the fact that many bacteria possess genes conferring resistance to several threats: resistance to environmental threats, resistance to competing bacteria and viruses, and resistance to antimicrobial chemicals and drugs. Having these genetic traits may be good for them, but it's not so good for us. When we treat diseases in humans and animals, we rely on powerful therapies, including antibiotics, to eradicate specific pathogens. But when antimicrobials are used, while susceptible bacteria can die off, as you can see in circle one, some bacteria harboring antimicrobial resistance genes, you can see they're colored in red, well, they're able to live and propagate, as you can see in circle two. Since bacteria move and exchange genes, these resistance genes can be passed to other bacteria, as you can see in circle three. And so in this way, resistant traits can be transferred to pathogens, which cause health problems in humans and animals. This antimicrobial resistance, or AMR, is a major global health and food safety threat. According to the CDC, in the United States alone, AMR accounts for one infection every 11 seconds and one death every 15 minutes. Therefore, my research attempts to address when and where does antimicrobial resistance pose a risk in microbiomes. So to answer this question, we use several approaches. First, we developed laboratory methods called targeted enrichment, which allows us to detect and isolate all known resistance genes in microbiomes. Second, we use sequencing and computational tools to reveal which resistance genes are actually present. Third, we use statistical methods to determine which resistance genes are more likely to be transferred among bacteria. Finally, we compile the information to produce a genomic risk profile. By profiling risk in this way, we will be able to disarm the microbiome and its contribution to antimicrobial resistance as a human and animal health threat.